Hi everyone, I'm Dr. Ye, currently an Associate Professor of Neurology in Taipei Medical University. Today is the first day of this semester. I am in charge of arrangement of this new course for international master students. The course is entitled as Biological and Clinical Perspectives of Human Disease. This course is to combine fundamental science and clinical medicine to introduce human diseases in many aspects, including oncological, infectious, cardiopulmonary, neurodegenerative, sleep medicine, and future therapies. Each lesson will provide an overview of human diseases by linking fundamental cell and molecular biology to clinical relevance and outcomes. We have arranged 16 lectures to illustrate current diagnostic and clinical treatment plan for patients with various diseases. The first lecture is delivered by me. Talk about the genetics in Parkinson's disease, including the genetic discovery, clinical overview, and the application on therapy. The following lectures will talk about research on several types of cancers and the application of immune and cell therapy, viral and parasitic infections, neuromuscular disorders, updated Alzheimer's disease therapy, and hot topic on microbiota and sleep medicine. I am very glad to invite my mentor, Washington University in St. Louis, Professor David Gartman. He is the world-known scholar. Is devoted to neurofibromatosis clinical and basic research for more than 30 years. He will give a lecture on October 27th. Due to time difference between Taiwan and the US, this online live lecture will begin at 9 p.m. Please save your time for the great opportunity. We don't have midterm or final exam. The assessment tool for the course is attendance and after lecture report. I will also remind you the online information. Please go to im.tmu.edu.tw for the information of the course. Okay, let's start the first lecture of the new course. The topic is Genetics in Parkinson's disease, from clinic to bench. This is the outline. Parkinson's disease was initially described by Dr. James Parkinson in his published article and essay on shaking palsy. He described 6 patients in London. They presented involuntary tremulous motion with lessened muscular power, in part not in action, and even when supported, with a propensity to bend the trunk forward and to pass from a walking to a running pace, the sense and the intellect being uninjured. Decades later, his name was used to designate the Parkinson's disease. Parkinson's disease is the second most common neurodegenerative disease in the elder population after the Alzheimer's disease. The prevalence is age-dependent. 0.5 to 1% of the population aged 65 to 69 years, and increasing to 1 to 3% of the population over 80 to years of age. The lifetime risk of developing the Parkinson's disease is 1.5%. In Taiwan, there are about 40,000 patients affected by Parkinson's disease. The cardinal motor manifestation of Parkinson's disease include TRAP, resting tremor, particular pure rolling tremor, cochlear rigidity while moving the wrist or arm joints. Brady or akinesia is shown as slow motion, loss of facial expression called mask face, and immobility of limbs. Postural instability on walking may cause fascinating gait or falls. This is a video clip demonstrating the tremor of hand and legs at rest in a 60-year-old lady. The other video shows slow motion of left hand and foot. Also reduce left arm swing on his walking in this gentleman. There are two main pathological findings of Parkinson's disease. One is the loss of dopaminergic neurons in substantia nigra pars compacta of the midbrain. The other is eosinophilic inclusion in neurons, called Lewy body, which is immunopositive to alpha-synuclein. The clinical motor symptoms of Parkinson's disease appear when at least 50 to 60% of the dopamine neurons are lost. The classical motor of basal ganglia pathophysiology in healthy and Parkinsonian state is illustrated in this figure. The input to basal ganglia from most cortical areas. Output is via GPI-SNR, which provides inhibitory input to the thalamus. The direct pathway expresses D1 receptors, while the indirect pathway expresses T2 receptors. In healthy condition, GPI output is modulated by competing influence from both direct and indirect pathways. In Parkinson's disease, death of substantia nigra pars compacta dopaminergic neurons results in overactivity in the indirect pathway and underactivity in the direct pathway, leading to heightening GPI output. In Parkinson's disease, loss of dopaminergic neurons in substantia nigra pars compacta cause dysregulation of GPI output to thalamus, then cortex, result in abnormal movement disorder. Several famous people were affected by Parkinson's disease. For example, Canadian-American actor Michael J. Fox. He achieved recognition as protagonist Marty McFly in the Back to the Future film trilogy on 1980s. Muhammad Ali was an American professional boxer, ranked as the best heavyweight boxer of all time. Adolf Hitler was an Austrian-born German politician. He initiated World War II in Europe by invading Poland on September 1st, 1939. Pope John Paul II was the head of the Catholic Church and the scion of the Vatican City State from 1978 until his death in 2005. The pathogenesis of PD is thought that complex interactions between genetic and environmental factors induce several cellular and molecular mechanisms, including ubiquitin proteasome dysfunction, mitochondrial damage, neuroinflammation, oxidative and nitrative stress, dopamine metabolism. Those mechanisms result in accelerating the age-related neurodegeneration. The PD phenotype appears while the dopamine neuron loss reach the threshold. The more influence of genetic factors contributes younger onset age of PD patient. Approximately 10 to 15% of people with PD have a family history of this disorder, and less than 5% monogenetic inheritance. The early study of heredity of PD was by Gauss in 1880. There was 15% of his patients has positive family history of PD. Dr. Mahjong in 1949 showed 41% of his 194 PD patients have positive family history. From-term studies, similar concordance of 10 to 20% for monozygotic and dizygotic twins, and the relative risk increased to sixfold for age onset less than 50 years. This figure illustrated key genomic discovery in Parkinson's disease. Alpha-synuclein SNCA is the first genetic cause of PD, which was identified in autosomal dominant Parkinsonian family in 1997 by Dr. Polymeropoulos. The mutation carriers had the same clinical presentation and pathological findings of Lewy body in the substantia nigra dopaminergic neurons, and the later SNCA protein was found as an important component of Lewy body. The next year, a group from Japan led by Professor Mitsuno identified the Parkin mutations are the major genetic cause of autosomal recessive juvenile onset PD. Subsequently, DJ1 and PIN1 were identified in autosomal recessive PD, whereas ROC2 was identified in autosomal dominant PD. Since PD is a complex disorder, monogenic cause account for fewer number of cases. The international GWAS had been launched in 2005, and several risk gene loci has been identified recently. Since PD is a complex disorder, monogenic cause account for few number of cases. Those gene loci identified from GWAS contribute the genetic risk factor for PD. This table summarizes the monogenic cause of PD, which account for 30% of familial and 3 to 5% of sporadic cases. The red ones are autosomal recessive inheritance. Patients with those mutations usually have earlier onset of PD symptoms. This is a list for susceptibility genes or loci for PD. Genetic architecture of Parkinson's disease shows the continuum of variants of different effect strength and allele frequencies. The size of the bubble roughly corresponds to the population allele frequency, color symbol, modes of inheritance, dominant as red, recessive as yellow, risk loci as green. The genetic risk for PD can be roughly divided into 3 groups. The group of high-risk genes contains monogenic PD genes. The group of common low-risk genes include more than 90 putative independent genome-wide meta-analysis based on more than 13,000 patients and 95,000 controls. The first identified gene causing Parkinson's disease is alpha-synuclein gene in chromosome 4q22.1. Essential was identified in autosomal dominant PD family in 1997 by Dr. Polymeropoulos. The mutation carriers have same clinical presentation and the pathological finding of Lewy body in substantia nigra dopamine neuron, and the latter essential protein was found as an important component of Lewy body. The second PD causing gene is Parkin gene in chromosome 6q26. The Japanese group led by Professor Mitsuno in Juntendo University identified the Parkin mutations are the major genetic cause of autosomal recessive juvenile onset Parkinsonism in 1998. In our 23 patients with Parkin mutation, the average age at onset is 28 years. And there are some characteristic clinical findings, including higher frequency of dystonia as its initial presentation, higher rate of levodopa-induced dyskinesia, and obvious sleep benefit. And the dopamine transporter image demonstrated symmetrically reduced uptake in basal ganglia in Parkin carrier. The clinical and genetic analysis of our 23 patients showed that the mutation type include homozygous deletion, compound heterozygous deletion, and mutation. Alternative allele deletions, heterozygous deletion, and mutations. Exon deletion were the most common type of Parkin mutations, especially exon 5 and between exons 2 and 3. The Parkinson, a hereditary early onset PD, is caused by mutation in PINK1 gene, which was discovered by Dr. Valente in autosomal recessive PD family in 2004. The clinical features of our 4 patients with PINK1 mutations showed that PINK1 patients had a typical phenotype with juvenile onset, benign course, and frequently with dyskinesias. And the dopamine transporter image demonstrated a rather even and symmetrically reduction of uptake in basal ganglia. Another important gene for autosomal dominant Parkinson's disease is LRRK2, LRRK2 mutations, which was identified by 2 groups, Payson-Ruiz and Zinprich, at the same time in 2004. The most important LRRK2 gene mutation is G2019S, owing to its high incidence in European countries, but this mutation was not found in Han Chinese. Two other genetic variants, G2385R and R1628P, were found in our population, but not in Caucasian. The estimated ancestor is founded from the era of Emperor Huang, around 4,000 years ago from now. Although it is not like the strong disease-causing effect of G2019S mutation, the younger age at onset was found in the LOG2 G2385R carriers. The PAR2G6 mutation was first identified in patients with neurodegenerative disorders with high brain iron by Professor Hayflick in 2006. Later on, the PAR2G6 was characterized as a loci for autosomal recessive PARK14 by Dr. Payson Ruiz in 2009. PAR2G6 mutations cause PARK14-linked young-onset Parkinson's disease and increase the risk for sporadic PD. I will show you our study from clinical to bench in the following section. Another gene for autosomal dominant PD is VPS35 mutations, which was also identified by Zimprich in 2011. His group also identified LOG2 gene. The recent study identified LRP10 genetic variant in several familial Parkinson's disease and dementia with Lewy body patients, including our two families. The first GWAS in Parkinson's disease is published in 2005. Some susceptibility loci were found. The meta-analysis of GWAS of more than 5,000 PD and 12,000 controls revealed that several sequence variants may contribute the risk for PD. Interestingly, SNCA and LOG are also found in high association with sporadic PD patients. This is a website posting the PD-related genes. This is my former PD genetic research team. Professor Qinsong Lu, Professor Hongli Wang, Professor Yichen Chen, Professor Qinqi Chiu, and me. The genetic spectrum of Parkinson's disease in our Taiwanese cohort shows Parkin is the most common genetic cause of young-onset PD patients with age onset less than 30 years, followed by PINK1 and PAR2G6 mutations. In sporadic Parkinson's disease, LOG2 G2385R account for 10%, followed by LOG2 R1628P in 7.4%, and GBA L444P in 2.8% of patients. This is a video clip of our PARK14 patient. It's a 25-year-old Taiwanese lady with compound heterozygous mutation of PAR2G6 gene. She presents dystonia and Parkinsonism, slow of movement. In our 2012 study, we identified 4 PARK14 patients with PLA2G6 mutations. The D331Y is the most common genetic variant. The patients had abnormal dopamine transporter scan and some cases had atrophy of cerebellum. In this study, we also found that a synonymous mutation caused by single nucleotide change at the end of exon 7. This mutation could result in an aberrant splicing by activating a cryptogenic splicing site on the exon 7 of PLA2G6 gene. Clinical features of PLA2G6 mutation carriers is listed in a table. One patient carried D331Y homologous mutation, and 2 patients carried compound heterozygous mutations. They had levodopa-responsive Parkinsonian, but also had some atypical features such as ataxia, dysarthria, psychosis, and dementia. In addition, we did genetic analysis of PLA2G6 variants in 956 sporadic PD and 800 controls. PLA2G6 D331Y mutation increased the risk of early onset PD with an odds ratio for early onset PD 3.68-fold. PLA2G6 belongs group 6 of phospholipase A2, also known as PNPLA9 or IPLA2 beta. Phospholipid is the major component of our cell membrane, essential for membrane remodeling and renewal. PLA2G6 is a calcium-independent phospholipase, hydrolyzes the SN2 ester bond in phospholipids, and highly expressed and serves as dominant PLA2 in brain. PLA2G6 mutations can cause 3 phenotypes: neurodegeneration with brain iron accumulation (NBIA), infantile neuroaxonal dystrophy (INAD), and Parkinson disease 14 (PARK14). PARK14 is also known as young onset dystonia Parkinsonian with onset age 4 to 37 years old. Clinical features are levodopa-responsive Parkinsonian and neuropsychiatric disorder. Cell biological process impact by PD-related genes lead to lipid alteration. Different PD-causing or risk gene, GBA, SYNJ1, and PLA2G6 directly control lipid metabolism in neuronal cells. PD-causative genes, LOCK2, PINK1, and Parkin, may indirectly control lipid metabolism, localization, or signaling by controlling vascular trafficking and lipid exchange between various organelles inside the cell. Finally, alpha-synuclein physiology is tightly related to lipid. Alpha-synuclein controls lipid metabolism. Alpha-synuclein localization and aggregation are strongly dependent on specific membrane lipid composition. And lastly, Lewy bodies are largely composed of alpha-synuclein tightly associated with lipid and membrane fragments. The aggregated alpha-synuclein is represented in here. In our in vitro study using neurotoxin-induced PD cell model, we found that Park14 mutant PAR2G6 inhibits an impaired enzyme activity of phospholipase A2. Expression of wild-type PAR2G6 but not Park14 mutant PAR2G6 attenuates rotenone-induced death of SHSY5Y dopaminergic cells. The immunofluorescence image study showed that Park14 mutant PAR2G6 is ineffective in preventing rotenone-induced loss of mitochondrial membrane potential and ROS production. Park14 mutant PAR2G6 failed to prevent rotenone-induced reduction of mitochondrial pancreas-1 activity, intracellular ATP content, and rotenone-induced increase of mitochondrial lipid peroxidation and cytochrome C release. These findings suggest PAR2G6 is important for maintaining mitochondrial function. In advance, we established a knock-in mouse model expressing common PAR2G6 mutation D331Y to study the molecular pathology and biomarkers of PAR2G6 and to develop potential neuroprotective treatment. Behavior tests include locomotor activity and POR tests are performed in wild-type heterozygous PAR2G6 and homozygous PAR2G6 D331 mice at different ages. Compared with the wild-type and heterozygous D331Y mice, the homozygous PAR2G6 D331Y knock-in mice exhibit reduced locomotor activity assessed by ambulatory count and increased time to descend the pole in the POR test at the age of 6 and 9 months, represent to the motor phenotype of Parkinsonism. The tyrosine hydroxylase TH immunostaining demonstrated that compared to age-matched wild-type mice or heterozygous mice, the number of Th-positive dopaminergic neurons were significantly reduced in the substantia nigra pars completa of PAR2G6 D331Y homozygous knock-in mice at the age of 6 or 9 months. 6 or 9 months old PAR2G6 D331Y knock-in mice also display a significant decrease in the number of SNPC cells. The established PAR2G6 knock-in mouse model can replicate the classical Lewy body pathology of Parkinson's disease at the age of 9 months. Lewy body in the substantia nigra of homozygous PAR2G6 D331Y knock-in mice at the age of 9 months was detected by performing immunocytochemical staining of anti-phosphoalpha-synuclein and anti-alpha-synuclein. Further immunoblotting analysis show an upregulated protein expression of alpha-synuclein, phospho-alpha-synuclein, and phospho-tau protein in the substantia nigra of homozygous PAR2G6 D331Y knock-in mice. In the neuromelanin positive putative substantia nigra pancreas competa dopaminergic neurons of wild type or heterozygous mice, mitochondrial morphology was intact, and the mitochondrial crystal had a regular arrangement. In contrast, disrupted structure of mitochondrial crystal was observed in the neuromelanin organelle containing putative substantia nigra dopaminergic neuron of homozygous Partridge D331Y mice. Compared to wild-type mice, the size of mitochondria was decreased in the substantia nigra of homozygous D331Y Partridge mice. The activity of mitochondria complex 1 and 3 was significantly decreased in the substantia nigra of Partridge D331Y homozygous knocking mice as compared with wild type. Intracellular ATP level was reduced in the substantia nigra of homozygous D331Y knocking mice compared to wild type or heterozygous mice. Overproduction of ROS was observed in the homozygous mice. The level of mitochondrial lipid peroxidation was upregulated in the homozygous D331Y Partridge mice. Cytosolic level of cytochrome C was increased in the substantia nigra of Partridge D331Y homozygous mice. Combined with previous ultrastructure findings and this finding, it indicates that homozygous D331Y Partridge mutation lead to abnormality in the ultrastructure of mitochondria and mitochondrial dysfunction. Western blot analysis showed that protein expression of active caspase 3, active caspase 9, or cytochrome C in the cytosol was upregulated in the substantia nigra of homozygous D331Y Partridge knocking mice at the age of 6 months. Protein levels of GRP78, IRE1, PERK, and CHOP were increased in the substantia nigra of Partridge D331Y homozygous knocking mice. Immunoblotting analysis showed that protein expression of Parkin or BNIP3 was significantly downregulated in the substantia nigra of Partridge D331Y homozygous knocking mice. These findings indicate that homozygous D331Y Partridge mutation causes activation of mitochondria at apoptotic cascade, induction of ER stress, and mitophage dysfunction. In this study, we established Partridge mutant model of Parkinson's disease for the drug screening. Injection of several Partridge mutant construct into zebrafish embryos. The frequency of spontaneous calling was reduced by D331Y or T572I injection. The injected embryos displayed reduced escape distance compared with the controls. Immunohistochemistry using tyrosine hydroxylase Th antibody showing that the reduced number of dopaminergic neurons following injection with D331Y T572I or R741Q construct. In addition, the dopamine level analyzed by HPLC was decreased by the injection of D331Y or T572I. Because the level of free fatty acid DHA 22.26 is decreased in lava injected with D331Y or T572I. We investigated the potential therapeutic effect of DHA, docosahexaenoic acid. In our study, supplement of DHA reduced the dopaminergic neuron loss and improved the motility defect in Parkinson mutant zebrafish, indicates that DHA can rescue the phenotype of Parkinson mutation in this zebrafish model. Furthermore, we assessed the effect of dietary DHA supplement in Parkinson D331Y homozygous knocking mice. Wild-type Parkinson and Parkinson homozygous knocking mice were administered DHA 35 mg per kilo per day from the age of 6 to 8 months. Upper panel showed 6 and 8 months old. Parkinson homozygous knocking mice displayed motor impairment as examined through the paw test, rotarod test, and horizontal ladder test. In contrast, DHA treatment nullified motor defect in 8-month-old Parkinson D331Y homozygous knocking mice, as shown in the paw test, rotarod test, and horizontal ladder test. Advances in molecular genetics over the past decades have been remarkable. DNA fortune telling. Genetic information could determine disease destiny from birth. Like the cartoon, fortune teller said, if you want the real lowdown, we'll need some of your DNA. And you cannot change a thing, as you are completely controlled by your genes. Google co-founder Sergey Brin has started a personal blog in September 18, 2008, and he went from describing his title choice to revealing some significant health news. He is at potentially high risk for contracting Parkinson's disease. The story started from that Brin's mother and aunt suffered from Parkinson's disease, in suggestive of a genetic link. Sergey Brin said he discovered he was carrying LAC2G2019S mutation after he ran a test provided by 23andMe, the genetic testing startup founded by his wife, Anne Wojcik. While the exact implications of the test result are not clear, Brin said it meant his chance of developing the disease compared with the average person was somewhere between 20% to 80% depending on the study and how you measure. But Sergey Brin is optimistic and positive. He said, "This leaves me in a rather unique position. I know early in my life something I'm substantially predisposed to. I now have the opportunity to adjust my life to reduce those odds." For the application of genetics in clinical medicine, there are several aspects including genetic decoding and genetic counseling, identification of risk factors, investigation of pathogenic mechanisms, establishing the animal models, and the development of therapeutic targets. We aim to detect before the symptoms onset, the gateway to turning back the degenerative process. To develop a pre-motor phase and neuroprotective therapy is our future. At the end, I would like to quote a description in Huangdi Neijing, the first Chinese medical textbook published in 2600 BC. He said, "Superior doctors prevent the disease. Medical doctors treat the disease before evident. Inferior doctors treat the full-blown disease. With the genetic knowledge, the era of preventive medicine and personalized medicine is coming." Thank you for your attention. If you have any question, please post it on the im@tnu or email to me. Thank you.